Now that we understand some of the designs that help us study development, we can start talking about specific periods of development. And we're going to start with prenatal development. Now, some of this is going to touch briefly on the side of more developmental biology as we start talking about what's happening as we move from conception through to birth. But we're also going to sort of look at the psychological side of things as well. So if you've taken a developmental biology, or if you are planning on taking developmental biology, you'll delve into a lot more of the biological side of this process. But we do need a very brief introduction to that to start understanding what's going on the psychology side of things, how our biology leads to our perception and our behavior and cognition and all of those cool things that we're going to get to talk about in this chapter. But on that note, let's start with the 3 stages of prenatal development. The first of these stages is the germinal stage. So this stage starts at conception, the moment that a sperm and an ovum or an egg cell form or fuse together to form a zygote. And a zygote is just the proper term for a fertilized egg. And this stage is going to last from conception up to 2 weeks' time. And during this time, the zygote is going to eventually attach to the uterine wall. And that happens towards the end of this stage at the 10 to 14 days mark. And as soon as that happens, we start moving into the embryonic stage. And this is going to be your 2nd to 8th week post-conception. At the beginning of this stage, 2 of our essential life support structures develop. This is the placenta and the umbilical cord. Now, the placenta is going to stick to the uterine wall, and it's going to be a membrane that's basically going to allow nutrients and different things from the mother's blood to pass into the embryo. And so the embryo is what we're now calling what in the previous stage we called a zygote. So as the zygote gets more nutrients and starts growing and dividing, we transition to calling it an embryo. And these nutrients that are transitioning from the mother to the embryo travel through the umbilical cord. So it's just going to be a cord that connects the embryo to the mother, containing lots of blood vessels, and it's going to allow nutrients to pass into the embryo. I'm also going to give you things like oxygen, very important for a developing ball of dense cells. And it's also going to transfer waste products back from the embryo to the mother. So there's a sort of an exchange of good things coming in and bad things going out to allow that embryo to continue to develop. With this constant influx of nutrients, those cells in the embryo are going to divide rapidly, and they're going to start specializing. So we can start seeing things like organs and systems beginning to form. So by week 8, you're going to see their heart start to form, and it's going to beat. The brain starts forming. You can start recognizing some facial features, things especially like the eyes. So natural progression there. This brings us to the 3rd and final stage, which is the fetal stage. So now we no longer call it an embryo, we're going to call it a fetus. And this stage lasts from 9 weeks post-conception right through till birth. We're going to see that continued development where their muscles start strengthening, their systems continue to develop. By 24 weeks, their eyes will begin to open, and by the time we hit 28 weeks post-conception, you're into that period of viability, where the child could survive birth. Now, while a fetus can survive once it hits that period of viability, their odds of survival after birth increase as you get closer and closer to full term, as opposed to being born preterm or premature sometime after that viability period. All right, now before we stop with the biology side of things, we're going to continue just a little bit more and talk about genetics and sex determination. So here we're talking about biological sex in terms of which chromosomes you have and whether that counts as male or female biologically. Now, from your biology classes, or even from 104, you may remember that humans have 23 chromosome pairs. So we have, actually, we can look at our karyotype here, where we have all of the different chromosomes that we have from 1 to 22 for our somatic or non-sex chromosomes. And then our 23rd pair has a couple of options. You can either have an X or a Y, depending on, well, your genetics. So in the case of females have 2 X's, so they'll end up with 2 of these larger chromosomes, whereas a male ends up with 1 X and 1 Y. And the Y chromosome in humans is actually what determines whether someone is biologically male or female. And that's because this Y chromosome contains something called TDF, which is testosterone determining factor. It's basically a gene that's going to initiate the development of the testes. And testes secrete androgens, so they're going to be secreting and releasing hormones that trigger the development of male characteristics. So during a critical period, in that 6 to 8 weeks post-conception phase, so during that embryonic stage, there's a period in which, if there is a Y chromosome, this TDF gene should trigger the production of androgens so that male characteristics develop. If there is no presence of androgens to trigger that development, if you end up with insufficient androgen activity, then you end up with the default female characteristics. So everyone starts as female, and then depending on the presence of specific hormones, some individuals will then end up developing male traits. Now that we've talked about how things are supposed to go during prenatal development, we're going to talk very briefly about a couple of environmental influences that can change or even cause problems during that development. So we're going to start talking about teratogens, and these are environmental agents that can cause abnormal fetal development. Basically, something in the environment that can negatively impact the growth and development of that zygote to embryo to fetus stage. And so an example of this is that there are things like drugs. There are a couple of drugs that are used to treat epilepsy or bipolar disorder that are known teratogens. And that actually raises a couple of ethical problems, because you run into the issue of, do you treat the mother for a potentially life-threatening disorder, or do you withhold treatment for the sake of the developing fetus? And that's quite the debate, and not everyone agrees on where we should fall on that issue. Other teratogens can be things like lead or mercury or radiation from your environment, even things like nicotine. Nicotine counts as a teratogen because it can cause miscarriage, premature birth, low birth rate, cleft palate, and some of those symptoms, those potential side effects that come from usually when a mother is smoking, can also be triggered if someone else in the household, say the father, is smoking. So it's not just things from the mother that can negatively influence the fetus, it can also be other environmental factors as well. And we'll talk a little bit later also about alcohol as another teratogen. Something else we should keep in mind is that things like sexually transmitted diseases can pass from the mother to the fetus. So things like syphilis or even AIDS and HIV can travel to the fetus from the mother. And depending on which disease that we're looking at, these can produce things like brain damage or blindness or deafness or all of the above, basically depending on the severity and what it is that we're looking at. So things can pass from the mother to the fetus despite sort of the filtering nature of the placenta and umbilical cord. And like I just mentioned, alcohol can also be a teratogen. And there is a whole spectrum of disorders called fetal alcohol spectrum disorders, or FASDs, that can arise when the mother consumes alcohol while pregnant. And these spectrum of disorders can involve a whole range of cognitive, behavioral, and physical deficits caused by that prenatal exposure to alcohol. And the intensity of these deficits can range from mild to severe. And this is actually one of those times where the amount that you're exposed to, the timing of what you're exposed to, a whole bunch of factors all interact. So it's very difficult to predict sort of if and when a fetus is going to be severely impacted or mildly impacted. And that's why it's such a massive risk to consume alcohol at any point during pregnancy, because it's very difficult to determine if there is a safe point to drink. Hence, of the advertisements and awareness campaigns to try and warn people of the dangers of drinking alcohol while pregnant. But one of the most well-known of these fetal alcohol spectrum disorders is fetal alcohol syndrome. And this specific syndrome involves a cluster of severe developmental abnormalities, things like intellectual disability, attentional and perceptual deficits, impulsivity, poor social skills, a whole bunch of things to consider. And interestingly, children who suffer from that fetal alcohol syndrome, they also have physical side effects. So their faces form a little bit differently. Their faces look smaller. And they also have issues with brain development. So on the top here, we have the brain of someone with fetal alcohol syndrome. And then on the bottom, we have a control brain of equivalent age, but someone who doesn't have fetal alcohol syndrome. So we have that underdevelopment of those who have fetal alcohol syndrome. So quite a dramatic difference. And this difference in brain structure is probably a fairly strong driving factor for all of those deficits that we talked about that are associated with fetal alcohol syndrome. And then to wrap up, we can talk about the last couple of environmental influences that the textbook mentions, and these are maternal malnutrition. So basically the mother doesn't have proper nutrition if they don't have access to lots of nutrients that they can then pass on to their growing fetus. The risks are miscarriage, premature birth, stillbirth, impaired brain development. So kind of logical things to go wrong if the fetus doesn't have enough nutrients and nutrition to grow and develop normally. And similarly, maternal stress, specifically having lots of stress hormones circling in her system, can also negatively impact the development of the fetus, again causing the risk of premature birth. But even after the child is born, they show signs of irritability. And even attentional deficits. So a child who's born to a more stressed mother is going to be more prone to being stressed out themselves. So if they are disturbed and start crying, it's a lot harder to calm the child of a stressed mother down to stop crying than it is to calm the child of a non-stressed mother, which is fairly interesting. So lots of different environmental factors can influence the child's growth and development before they're born.